Segment 2 begins with the section titled Attention helps us to search a cluttered world for specific patterns. Attention helps us to systematically scan the world where we first find a specific item to help us find a specific item in a sea of distractors. Feature search is a search for an item in which the target pops out right away because it possesses a unique attribute. This allows us to easily pick out something, for example, like a red coat in a sea of black coats. More difficult, though, is the conjunction search, which is search for an item that is based on two or more features that together distinguish the target from distractors that may share some of the same attributes. This performance is related to the number of distractors present, and it makes us coordinate multiple cognitive feature maps. Feature integration theory is the idea that conjunction searches involve multiple cognitive feature maps, which are overlapping representations of the search array based on individual stimulus attributes. Simple pre-attentive stimulus attributes guide our scanning of the environment, with higher order processes coordinating the binding of features, leading to conscious perception and selection. The binding problem, though, is the question of how the brain understands which individual attributes blend together into a single object, when these different features are processed by different regions in the brain. Bottom-up processing is a process in which lower-order mechanisms like sensory inputs trigger additional processing by higher-order systems. There may be no conscious awareness until late in the process for bottom-up processing. Top-down processing, then, is a process in which higher-order cognitive processes control lower-order systems, often reflecting conscious control. We uncover the details of the brain's attention mechanisms based on the consequences of attention in the brain and the mechanisms of attention. Temporal resolution is the ability of imaging technique to track changes in the brain over time. This includes electrophysiological approaches, whereas spatial resolution is the ability to observe detailed structures of the brain using fMRI. These two are employed to uncover the details of the brain's attention mechanisms. The next section is titled Attention Affects Functioning of the Brain. We usually see how attention affects the function of the brain through EEGs, but it's usually hard to see the exact brain regions working together during a specific task. So the way that we deal with this is that we make the participants do the task over and over again to average all the EEGs to find a distinctive pattern of synchronicity during said task. This leads to these averaged EEGs that happen over and over again are called event-related potentials, or ERPs, which are average EEG recordings measuring brain responses to repeated presentations of the stimulus. The next section is titled Distinctive Patterns of Brain Electrical Activity, Marked Shifts of Attention. The study of auditory attention uses dichotic tones where we attend to one and ignore the other. Usually, half the participants ignore the left ear and half the participants ignore the right ear stimuli. ERPs for the attended stimuli signify that there's a larger amplitude than when the same stimuli at the same ear were not being attended to. This is called the N1 effect, which is a negative deflection of the ERP occurring at 100 milliseconds after the stimulus presentation that is enhanced for selectively attended input compared with ignored input. Based on this, we have selective attention acting on neural mechanisms to enhance processing of information from that ear. The P3 effect, then, is a positive reflection of the ERP occurring 300 milliseconds after the stimulus presentation that is associated with higher order auditory stimulus processing and late attentional selection. We usually use the P3 as a consciousness marker. During visual studies, valid trials show a P1 effect. Invalid trials being there is a cue that signifies where the stimulus will be, and it is correct where the stimulus will be. This leads to the P1 effect, which is a positive deflection of the ERP occurring 70 to 100 milliseconds after stimulus presentation that is enhanced for selectively attended visual input compared with ignored input, and it usually enhances following the N1 component. This signifies an attentional bottleneck because attention boosts activity of the occipital sensory processing. During invalid trials, where a cue signifies that a stimulus will be presented at a certain area, but then the stimulus is presented at an opposite area, do not show the visual P1 effect, which is an increase in amplitude of the P1 component of the ERP that occurs for stimuli that are the focus of attention. It is more associated with longer latency ERPs because it takes a longer time to find the stimulus when we direct our attention to where the cue tells us to go, usually about 400 milliseconds after the stimulus. Directing attention to find a particular target in a sea of distractors leads to an enhancement of a subcomponent of the N2 at the occipitotemporal sites. This leads us to maintain fixation on a central point. The test for this forces us to maintain fixation on a central point. Then a stimulus array is presented briefly, and then visual search for the stimulus needs to be rapid. PD then is a large positivity over the occipital region associated with ignoring attention-grabbing distractors. Neural mechanisms for visual attention are plastic, which is evident mostly in video gamers. The last section is titled Neuroimaging confirms the anatomical foci of attention show augmented processing. PET and fMRI are a consequence of attention show that a consequence of attention is an enhancement of activity of the brain regions that process key aspects of target stimuli. We use ERP to decide when to collect fMRI, which leads to anatomically distinct regions of enhanced activity in the visual cortex that correspond to the spatial locations to which participants directed their covert attention. This shift in attention is followed by a locus of enhanced activity shifts. Also, when we divide our attention, there's usually two areas of activity. The effect of attention on neural activation is evident also in subcortical visual structures. fMRI enhancements are shown in the superior colliculi and the lateral geniculate nucleus. Later stages of the visual system show physiological effects of attention. Fusiform face areas shows an ERP seen in cortical region responsible for face processing, and parahippocampal place area are responsible for processing locations. And that is Chapter 18, Segment 2.